Okay, good morning, everyone. It is 11 o'clock on my watch. Welcome back to the second session of today. The early morning's talks were very interesting, and I'm also very much looking forward to the second session of this morning. So the first speaker of this session is Mr. Chipang Ndaba. Chipang, if you could share your screen, please, and then we can go ahead with you. So the two speakers of this session are both from the University of Johannesburg. And the first talk given by Chipang Ndaba will be about overexpression, purification, and characterization of the HSP7014 protein towards the discovery and development of new anti-cancer compounds. Okay, Chipang, are you there? I've seen you just now.
Yeah, he's just trying to get on.
Good morning. Hi, I'm here.
Okay, excellent. We can see your screen. You can switch to full screen mode.
Okay.
Okay, excellent. So when you're ready, you're welcome to start. So I'll give you about 25 minutes, and then we'll have 5 minutes for questions after that.
Okay,
Wait, are we going out of order? I thought Chipang is the 11 o'clock speaker, right?
Yes. We are starting with Chipang. So Kagiso, you're the second speaker. Yeah, but it is Chipang's talk, so
okay.
Yeah, you can start when you're ready.
Okay, good morning, everyone. Today I'll be presenting my research project titled The Overexpression, Purification, and Characterization of the HSP70.14 protein towards the discovery and development of new anti-cancer compounds under the supervision of Prof. Kapo and co-supervision of Dr. Masamba. Now, cancer refers to the development of abnormal cells characterized by the uncontrollable cell growth and additional ability to infiltrate and destroy normal body tissues. Several cancer types exist, which are named according to the location in which they are first described. Several of these cancer types have been depicted in the presented slide, namely skin cancer, hard palate cancer, liver cancer, as well as breast cancer, all of which display uncontrollable cell growth rates. Now, cancer remains one of the leading causes of death worldwide, with over 18 million cases reported by 2020 and over 9.5 million cancer-related deaths. These numbers are expected to further increase to over 30 million cancer cases and over 16.5 million cancer-related deaths by the year 2040. And in South Africa specifically, the data obtained from the World Health Organization reports a total of over 100,000 cancer cases and over 50,000 cancer-related deaths, with the most prevalent cancers being breast cancer in women and prostate cancer in men, respectively. Several therapeutic approaches have thus been developed over the years, with chemotherapy being the most widely used treatment approach, which uses drugs to inhibit cell division. This has negative effects, however, on cells that naturally undergo rapid turnover, such as skin cells, hair cells, as well as bone marrow. Hormone therapy, on the other hand, typically acts to inhibit androgen and estrogen action in men and women, respectively, whilst radiation therapy utilizes radioradiation to initiate DNA damage and ultimately promote cell death. This has to be minimally used and carefully set up, however, as DNA lesions may lead to the accumulations of more mutations and the development of more cancer cell types. Whilst immunotherapy uses the body's own immune system to bolster the immune strength in the defense against cancer cells. This is, however, non-specific in the case of the innate immune system and thus leads to the loss of normal cells as well. And lastly, targeted therapy inhibits the promotion of tumors by inhibiting specific molecules that have been implicated in driving tumorigenesis. One of the molecules or proteins that have been implicated in driving tumorigenesis is the HSP70.14, which has been implicated in a paper by Capo and colleagues in 2012 to be involved in driving tumorigenesis. Now, HSP70.14 is a heat shock protein that has been seen to be upregulated at the onset of cancer. Now, heat shock proteins are proteins that are regularly expressed in cells in response to intra and extracellular signals such as heat and metabolic disruption. Heat shock proteins are responsible for protein refolding as well as guiding proteins to protein turnover. Heat shock protein 70 is the most widely expressed heat shock protein in the presence of heat stress. It is composed of an N-terminal domain, a substrate binding domain, as well as a C-terminal domain. HSP70.14 is a variant of the HSP70, displaying similar structural characteristics to the HSP70, the only difference being that the HSP70.14 lacks the C-terminal domain. This leads to the aim of my research, which is to computationally and biophysically characterize the HSP70.14 protein towards the discovery and development of new anti-cancer compounds, according to the following objectives, which is to perform computational studying using modeler to predict the structure of the HSP70.14, to recombinantly express the HSP70.14 protein in E. coli cells, and to subsequently purify the protein using affinity chromatography, followed by biochemical characterization using FTIR and Raman spectroscopy. The objectives of the project will be achieved according to the following methodology. Firstly, in silico protein characterization will be performed. This will be followed by in vitro studies. Firstly, transformation of the protein construct of interest will be transformed into E. coli cells. Expression screening will subsequently be conducted, followed by large-scale protein expression and affinity purification. The protein will then be subjected to biochemical characterization using FTIR as well as Raman spectroscopy. The HSP70.14 was then modeled and viewed in chimera and displays secondary structure elements composed of 2 alpha helices as well as 3 antiparallel beta strands. The Ramachandran plot used to validate the model structure is used to assess the stereochemical parameters of the protein. Amino acid residues are allocated into 4 regions, namely the allowed regions, the most favored regions, the additionally allowed regions, the least favored regions, and the disallowed regions. Now, from the generated plot, over 94% of the residues are allotted into the most favored regions and the additionally allowed regions. And for Marco Vulcan in 2018, at least 90% of the residues are required to be allotted in the aforementioned regions in order for the model to be deemed acceptable. The Q-min-Swiss model was also subsequently used to validate the model protein, and a Z-score of in the region of 0.5 was obtained. And from Benkert in 2008, a score in the region around 0.5 indicates a good quality model. The kite and Doolittle plot is used to give indication on the overall hydrophobicity of the protein based on the individual hydrophobicity of the amino acid residues that are present in that protein. From the generated plots, residues 1 to 20, as well as residues 80 to 100, all display a score of above 0, which represents hydrophobic regions of the protein. Majority of the amino acid residues, however, display scores of below 0 and thus represent hydrophilic regions of the protein, which are responsible for interacting with the surrounding polar solvents. The depicted string protein association network depicts.
Hi,
the
Silvana. This is
proteins that
Lawrence.
HSP 70.14 interacts with in cellular.
I wonder if this is a good time to bring up the science forum in South
The
Africa.
program string was used to depict the 10 other cellular proteins that the HSP 70.14 interacts with in cellular pathways.
Okay. All right. I think I missed the
The
sign.
generated string
Sorry, I'm
depicts
multitasking here.
that
So this morning.
HSP 70.14 interacts closely with 10 other cellular proteins, with several of these interactions showing that HSP 70.14 interacts closely with the back family of proteins, which have been reported to serve as adapter proteins by forming molecular complexes with molecular chaperones such as the HSP 70.14. A model was subsequently proposed that HSP 70.14 interacts closely with the cancer-associated BAC2 protein. This hypothesis was further supported by a recent article by Johnson and Pestwicki in 2022, in which they reported that the BAC family of proteins are pivotal regulators of eukaryotic HSP 70 complexes. The BAC2 was also thus subsequently characterized in silico, and the generated model in Chimera displays that the BAC2 model protein contains 2 alpha helices. The Ramachandran plot further validates the model protein by displaying over 96% of the residues allocated in the most favored regions, as well as the additionally allowed regions. The Q-min-Swiss model of the BAC2 protein also shows a Z-score of just above 0.5, which indicates that the model protein is also a good quality model. And the kite and Doolittle plots generated from the BAC2 protein displays that majority of the amino acid residues present in the BAC2 protein display a score of below 0, meaning that majority of the protein is hydrophilic. Molecular docking was subsequently generated and run using the HDoc web server, whereby the highest ranked model with the lowest GOP docking score of minus 29,367 was generated. In the depicted slide, the BAC2 is depicted in purple, whilst the HSP 70.14 is depicted in light blue. On the right side of the screen, several of the amino acid residues that are hypothesized to be involved in the interaction are also depicted, with some of these residues being leucine, arginine, as well as phenylalanine. The HSP 70.14 sequence was also run on the program iTaser to obtain ligands that have a C-score that is closest to 1. Now, a single compound was obtained, which is depicted as C1 in the presented slide, in order to compare and contrast to already existing and currently used pharmaceutical drugs in the treatment of cancer. Now, the ADMET table generated in the presented slide shows that the compound has a total molecular weight of 242 daltons, which is less than the currently existing therapeutic compounds and less than the recommended size of 500 daltons. The compound is also reported to be water soluble and displays high GI absorption, as well as a permeance to the blood-brain barrier. It also observes the Lipinski rules and shows the total bioavailability score of 0.55 and the synthetic accessibility of a total score of 2.26. This compound could thus be explored further in the continuous development of new novel anti-cancer therapeutics. In vitro studies were subsequently conducted to validate the preliminary bioinformatic results, as computational work only provides and only serves as the foundation for structure-based drug discovery. The following presented slide shows the result obtained for transformation of 4 E. coli cell lines. Firstly, the JM109 E. coli cell line was successfully transformed and E. coli bacterial colonies grew. However, there was no efficient protein expression of the HSP 70.14 protein, which is a 16 kilodaltons protein. Similarly, no efficient protein expression was obtained using the codon plus E. coli cell line. In comparison, bacterial colonies grew on the T7 agar plates with efficient protein expression observed in the T7 E. coli cell line. However, the bands were not distinct and compact enough in comparison to the top 10 E. coli cell line, which displayed the more compact and distinct band, which was subsequently the reason why it was chosen for downstream large-scale protein expression. The depicted and presented slide of the expression screening and large-scale protein expression gel displays that the HSP 70.14 was successfully and efficiently expressed in the top 10 E. coli cell line, particularly using colony 2 of the E. coli cell line. Following large-scale protein expression, a gel was run to check and confirm for the solubility of the protein with the protein observed in the cell lysate, which is depicted as CL in the presented slide, whereas there was no protein expression in the pellet depicted as P in the presented slide. This confirmed that the protein was indeed soluble and could thus be subjected to purification. The protein was then subjected to protein purification, but only small amounts of the protein were able to be purified. Large quantities of the protein were still unable to be purified, which may be due to the histidine tag of the protein constructs being located into the interior core of the protein and being unable to interact with the nickel on the affinity column. Nonetheless, the small amount of protein that were obtained from purification were used for structural characterization. Now, the results from the FTIR spectroscopy reveal a significant trough at a wave number of 3,310 per centimeter. Now, this trough is indicative of the presence of a hydroxyl group. The second significant trough observed at a wave number of 1,635 per centimeter is indicative of the presence of carbonyl group. Now, these results further validate the results obtained from the Kite and Doolittle plots, which suggested that the majority of the protein is hydrophilic. For Raman spectroscopy, the region of wave number 500 to 1,500 per centimeter is known as the fingerprint region of the protein, which is unique for every biological molecule. Now, from the generated Raman spectroscopy, a significant peak is observed at a wave number around 1,000 per centimeter, which is indicative of carbon-carbon symmetric stretching. A second peak is also observed at around 1,400 per centimeter, which is indicative of carbon-hydrogen twisting vibrations. Whilst another significant peak outside the fingerprint region of the protein at around a wave number of 3,000 per centimeter is observed. Now, this peak is indicative of high frequency carbon-hydrogen vibrations. Thus, in conclusion, urgent interventions are constantly required in the age of reduction of cancer mortality cases observed worldwide. The identification and investigation of proteins that are essential in driving tumorigenesis provide insight towards the advancement of current existing cancer therapeutic approaches. HSP 70.14 protein structure determination thus provides the initial basis for the development and discovery of novel cancer therapeutic drug compounds, and future studies will thus entail more comprehensive interaction studies between the HSP 70.14 and BAG2 protein using isothermal titration calorimetry as well as microscale thermophoresis. The results from the MST and the ITC is hoped to provide the basis for the development and structure-based discovery of new protein-protein interaction modulators whose advances and successes have been reported and well described in a paper by Mabonga and Kapo in 2019. These are some of the references that have been used throughout the project. I'd like to acknowledge all the contributing members to the project, and thank you all for your time.
All right, thank you very much, Chipang, for very, very nice work. It's a very good presentation, excellent work. So the floor is open for questions. We have plenty of time for questions. Trevor, I see your hand is up. You can go first.
Hi, Kagiso. That was very interesting talk. I had several questions that occurred to me. The one is, why did you use modeler in preference to AlphaFold?
Okay, well, my co-supervisor was helping me with that, and it seemed to be the one that she's most still familiar with at the moment. So that's the reason why we chose to go with Chimera at the moment. Although we are obviously currently looking on moving to programs such as AlphaFold.
Okay. And then the second question is HSP 70.14 a monomer when it's active in the body?
Yes, it is active as a monomer.
It is active as a monomer. And this is there a crystal structure of this compound?
Not as yet.
Okay. And have you been able to observe experimentally the interaction between HSP 70.14 and BAG?
No, we have not gotten to that yet. That's actually where we are heading with this project. With my master's work, we are looking into wet lab studies and actually experimentally looking into the wet lab interaction between the BAG2 and the HSP 70.14.
And how would you do that?
I'll either use ITC or MST. So I'm gonna express both proteins and purify them and then use either isothermal titration, calorimetry, or microscale thermophoresis.
It's too small a difference for gel filtration chromatography, is it?
Sorry, I didn't quite get that.
It's too small a difference for gel filtration to be seen by gel filtration. Or perhaps non-denaturing electrophoresis.
Are you asking, is the protein too small to be observed using gel filtration?
The interaction.
Yes.
Could you observe the interaction using gel filtration chromatography, or perhaps even band shift in non-denaturing electrophoresis?
Oh, I think that would be the case. I think you wouldn't be able to observe it using gel filtration, which is why we're also validating the interaction using also FTIR and Raman spectroscopy.
Does the FTIR and Raman spectroscopy enable you to see the interaction?
Well, it just shows you it doesn't give you the stoichiometry of the interaction, which is why we still have to do MST or ITC, but it does give an indication on whether there are any changes between the two proteins, which might be indicative of any interaction between the two proteins.
Okay, thank you.
Paul Kapo, I see your hand is up. Do you want to make a remark or a question?
Yes, I want to make a remark. I think I want to actually agree with what Trevor has actually pointed out, which are very, very important, because that is where we are heading with this project. And what Trevor has pointed out as regards the interactions is that the first thing is that there's no crystal structure of HSP 70.14 yet. I've not seen that yet. I don't know if it exists somewhere, but to our knowledge, there hasn't been that structure yet. We've got other HSPs, HSP 70.1, HSP 70, HSP 90, and so on. But as at the last time we checked, we haven't seen that. So our idea with this work is to actually validate our hypothesized interaction, which we think exists and which in silico we have seen that it does exist. But where we're going actually is that to now do the structure of the protein complex, we actually want to know, number one, three things. The first thing we want to know is that does that interaction actually exist? And that we actually will want to use the methods he has mentioned. We've got experience with MST and ITC. We have experience with that. We've got some publications which we've actually published using those two methods, ITC and MST, especially MST, which is microscale thermofluorescence. The second thing that gives to you is that it tells you the stoichiometry of how much of each protein do you need to form the complex. And then also it tells you also the strength of interaction, because we believe that for drug discovery, you basically want the two proteins to, to interact, and then you want to look for a disruptor of this interaction. So you need to know if the two proteins interact, what is the strength, and MST, you're able to get the MST via the KD. So that is where we are heading. Already we've got the second protein also expressed. We just need to purify and actually do this, confirm the interaction in the lab. So that is where we're heading for now. The issue with AlphaFold and using AlphaFold and Chimera modeler, I think it's all about what we are comfortable with. I think, yes, AlphaFold for sure will surely give us, but I read a paper recently. I just forgot the authors now. Recently, I think I can share that paper with Trevor at the end of this presentation, which talks about, I mean, a lot of the predicted structures which AlphaFold gives you, I think the huge percentage were actually found not to be completely, don't want to say correct, but some part of the secondary structures are missing. So I have that paper which I just stumbled upon recently. So that didn't give me that confidence to say we should go via the AlphaFold route. So I just said, let's basically go through what we know works, the manual Chimera modular part, and that is what he did with the co-supervisor. So I can still engage with Trevor at the end of the day, and obviously we're going to do the structure with Trevor anyway. So I'm happy he's asking these questions now. Thank you.
Thank you very much, Paul, for the insightful remarks. It's exciting to learn about your prospective projects, the way that this project is going. And could I ask you to post the URL of the article that you have referred to about the inaccuracies of AlphaFold in the chat?
Yeah.
I'm sure that many other participants would also be interested.
Okay.
Okay, thank you very much for those remarks. Okay, we have 2 minutes for a last question, if there's any burning question. Okay, if not, let's thank the speaker again. Thank you for a very nice presentation. And then we can move on to the next speaker. I understand that these two speakers are from the same research group and using the same account. So the first speaker was Pang Daba, and now Kagiso Motshedi is going to give a presentation on recombinant expression and biophysical characterization of NAD binding domain of S-Manzoni glycerol glycerolaldehyde 3-phosphate dehydrogenase. So Kagiso, you can share your screen when you're ready.
Okay.
Okay, I can see your screen. Please switch to full screen mode.
Okay. Can you see it now?
Yes, that's good. Okay, so the floor is yours.
Oh, okay. Good morning, everyone. My name is Kagiso Mtlatledi, and I'm going to be presenting my project titled The Recombinant Expression and Biophysical Characterization of the NAD Binding Domain of Cytosoma mensoni glyceraldehyde 3-phosphate dehydrogenase under the supervision of Prof. Kapo and co-supervision of Dr. Masamba. This is my presentation outline. I'll be taking you through the introduction all the way to the conclusion. Cystosomiasis is a chronic parasitic disease that is caused by cystosoma worms. It affects over 260 million people, with 800 million people at risk of infection. This disease is widely spread throughout the globe, with 3 major species that are affecting humans being Cystosoma japonicum, Cystosoma hematobium, and Cystosoma mensoni, with Cystosoma mensoni being more prevalent in South America, Africa, as well as Asia. This disease causes great morbidity and mortality. It is responsible for the death of over 280,000 sub-Saharan Africans annually. Infection begins when Secariae released from the snail intermediate host penetrates the human host skin when the human comes into contact with contaminated freshwaters. During this stage, the infected individual will experience Katayama fever, which is characterized by macular rash, abdominal pain, cold sweat, as well as diarrhea. These Secariae will then undergo various processes to become cystosomuli within the human host, and these cystosomuli will then travel to the liver of the human host, where they mature into adult worms that mate and reproduce. These eggs will then be released through defecation back into freshwaters, while others are retained within various locations within the body. And this is when the infected individual will experience the chronic infection, where they will experience bloody diarrhea, ulcerations, and also an increase in susceptibility to cancer and HIV. Ever since its discovery in the 1970s, praziquantel has been the only drug used to treat cystosomiasis, since it is cost-effective and readily available. However, the drug fails to kill the juvenile worms, and it doesn't prevent reinfection. The other drawback of praziquantel is that there are cystosoma strains that are resistant to this drug, which were first identified in Kenya and Brazil. However, these resistant strains are now found worldwide. Therefore, there's an emergence to explore other druggable targets for the development of new anti-cystosomal therapeutics. In a paper by Gokoi and colleagues, 2020, it was identified that proteins are possible drug targets for the discovery and development of new drugs to treat cystosomiasis. And one of the proteins that are identified in the paper is GAPDH. This protein plays a crucial role in the glycolytic pathway as it catalyzes the conversion of glyceraldehyde 3-phosphate to 1,3-diphosphatoglycerate using its NAD binding domain. This will then lead to a release of NADH, which is subsequently used in the electron transport chain in order to release hydrogen ions, which are then used in the subsequent complexes to produce ATP, of which the cystosoma worms use to drive their life cycle, such as production of eggs and maturing into adult worms. This means that this NAD binding domain plays a crucial role in the survival of these cystosoma worms within the human body. Therefore, this project aimed to recombinantly express and biophysically characterize the NAD binding domain of GAPDH protein of Cytosoma mensoni to provide the structural basis for the future discovery and development of new drugs to control cystosomiasis. These are some of the objectives that we use to achieve the aim. And for the methodology, firstly, in silico characterization of the protein was conducted to determine its 3D model, as well as validate the structure using the Ramachandran plots and the Cumin-Swiss model. The wet lab studies commenced by transformation of bacterial cells with the NAD binding domain plasmid in order to grow colonies, and random colonies were then picked for the small scale expression, and the best expressing colony was then used for the large scale expression in order to produce large quantities of the protein, which were then subjected to affinity chromatography for purification. Lastly, the purified protein was subjected to biophysical and spectroscopic characterization using FTIR and Raman spectroscopy. These are the in silico characterization results of the NAD binding domain that were obtained. The model structure of the protein showed that it contains 6 beta sheets and 4 alpha helices, which coincides with the findings of Lex 1995, who found that all NAD binding domains contain 2 units of beta alpha, beta strands. And this model was then validated using the Ramachandran plots, where it was found that 93.6% of the residues were in the most favorable regions, with no residues in the disallowed regions. And therefore, it was deemed as an acceptable model since it did not violate any stereochemical parameters. The Cumin-Swiss model was used to further validate this model protein, and was found that the protein had a Z-score of less than 1, which meant that this was a good model. Kite and Doolittle plots was used to determine the hydrophobicity of the protein, and was found that most of the residues of the protein were below 0, which meant that it contained mostly hydrophilic residues, and therefore the protein was hydrophilic. Since proteins do not function alone, but through protein-protein interactions, the string protein-protein interaction network was used to determine other interacting partners of GAPDH, and it was found that triphosphate isomerase highly interacts with GAPDH through its NAD binding domain. According to Bergard and colleagues 2007, proteins that perform similar functions and which are found in the same pathway are most likely to interact with each other. Both of these proteins are also located on the tegument of the cystosoma worms, where they elicit an antigen-antibody response, which helps in aiding the worms to survive in the human's immune system. These results then led to the expression and characterization of the TPI protein. It was found that the homology model of the TPI contained 8 alpha helices, which cover the 8 beta sheets, forming what is commonly known as the TIM spiral. This structure was then validated using the Ramachandran plots, where it was found that 94.6% of the residues were in the most favorable regions, which then deemed the structure as acceptable. The Cumin-Swiss model also showed that this homology model was a good model since the Z-score was less than 1. The Kite and Doolittle plots showed that this protein is hydrophilic, just like the NAD binding domain, since most of its residues were below 0. As mentioned before, that proteins do not function alone, but through protein-protein interactions, it was important to elucidate if these two proteins do in fact interact, and using molecular docking, the two proteins were then docked using H-dock, and the best model was chosen based on the negative docking score, which was minus 204,000, and it was found that these two proteins do in fact interact, and the residues that are involved in this interaction were also identified, which were found to be leucine, valine, serine, tyrosine, to name a few. However, it is important to keep in mind that this is only a prediction, and therefore wet lab studies should be conducted to validate this interaction between the two proteins. The Swiss ADME was used to determine compounds that could disrupt this protein-protein interaction to solve as an alternative avenue to solve the problem of drug resistance in cystosomiasis. And there was one compound that was identified, which is AK1, which had a C-score of 0.04, while others were lower. This compound was found to be highly similarity to praziquantel, which is the current drug that is being used. It is soluble in water, and it is also highly absorbed in the gastrointestinal. This drug also showed that it permeates the blood-brain barrier, and it doesn't violate the Lipinski rule of 5, which meant that this drug could be already active in humans. The bioavailability score of this compound is found to be 0.55, which is the same as praziquantel. The bioavailability score is supposed to be between 0.25 and 1 in order to show that it can cause a pharmacological response. Lastly, the synthetic accessibility of this compound was found to be 2.26, which is less than praziquantel, and the lesser the synthetic accessibility is, which is closer to 1, the more it will be able to synthesize this compound from the lead molecule in order to discover other drugs to treat cystosomiasis. These are the experimental lab studies that have been obtained so far to validate the in silico characterization results. Transformation of the Biom-109 cells using the NAD binding domain plasmid was successful, as seen in plate A, and 4 colonies were then picked from this plate to be used for the expression screening. As seen on the gel, the odd-numbered lanes are the uninduced samples, while the even-numbered lanes are the induced samples of each colony. The gel showed that in lane 7 and 8, there was more expression, and therefore sample 4 was then used for the large-scale expression. This large-scale expression was combined with harvest time optimization, and it was found that in lane 5 and 6, there was more protein produced, which was harvest time of 12 hours, and therefore these conditions were then used to obtain large quantities of the protein. The protein was then subsequently purified using the GST column and then characterized using the following techniques. FTIR showed that there was a significant peak at 1,638.14, which is indicative of amide white bands, and it indicates that there is the major element, which is beta sheets, which was found to be an element in the homology model of the NAD binding domain. Lastly, Raman spectroscopy showed 3 significant peaks, which were peak 1, 2, and 3, as seen on the graph, and which are indicative of amide 1, 2, and 3 bands, respectively. And according to Regula and colleagues, 2010, these amide bands are indicative of alpha helices and beta sheets, which were present in the homology model of the protein. In conclusion, the GAP-DH NAD binding domain of cytosol and SONI was successfully expressed, purified, and characterized. The in silico characterization of the biophysical characterization of the protein revealed that this protein contains alpha helices and beta sheets. Trans-phosphate isomerase was also identified as an interactive partner of GAP-DH through its NAD binding domain. Currently, the TPI protein is being expressed and will soon be purified and characterized. The characterization of the interaction will also be conducted using MST or ITC in order to determine the thermodynamic parameters of this interaction, such as the binding strength as well as the stoichiometry N. Therefore, the results obtained from this study are expected to form the structural basis for the future discovery and development of new drugs to treat schistosomiasis. These are some of the references that were used for this study. These are the individuals that I would like to acknowledge, and thank you very much.
All right, thank you very much, Kagiso, for a very nice talk, very interesting content. It's great science that you are doing. Okay, so the floor is open for questions. There is plenty of time for questions. So please raise your hand if you would like to ask a question, or since we are relatively few participants, you can also unmute yourself and just speak.
Good morning. Hi, it's Wolf-Dieter Schubert here. I would just like to ask about your modeling. So does your model support, or from your complex, can you infer that this might be the correct complex? I mean, either is there an existing complex that you can compare it to, or does it provide the expected dimensions? I presume this is about electron transfers, kind of. So is there good distance between cofactors so that it would make sense? So have you validated on a structural basis is my question, probably.
Oh, will the structure be validated?
No, the question is, can you infer from by looking at the structure that it actually makes sense the way you have modeled it? Have you looked at the structure, kind of have you analyzed the distances, etc.? Not just the amino acids, I think that's important as well. Kind of that's what I saw you did, but kind of looking at the structure as a whole, does it make sense?
Oh, no, I haven't really analyzed the structure, but then since I think the distance is supposed to be 4 degrees Armstrongs, if I'm not mistaken, so that is what the software uses. It tries to keep the distances to 4 degrees Armstrongs.
Yeah, okay. No, I mean between the different proteins, kind of they presume the 2 proteins that you've complexed, kind of are they in the correct orientation relative to each other so that they can do whatever they need to do, they hand over substrates or electrons What are they doing? Kind of, does it make sense?
Oh, I didn't really look much into that.
Okay. Okay, so you should perhaps kind of just look and see whether it actually makes sense internally. Normally, because the thing is, when you actually
You are muted, Bob Dieter.
Hello.
It's gone.
I think we have lost him.
Yeah, and I'm actually taking notes from what he's saying.
Yes.
Yes, I think while we are waiting for Wolf Dieter to come back, if there are other questions, please go ahead. Now, maybe a question from my side. So this is completely outside of my field of expertise, but as far as I know that the GAPDH is an enzyme that has been, well, whose structure has been known for quite some time. So the differences that you are investigating, have you actually compared the different, like the structures, experimental signals, if there are, Raman, all of that? Have you directly compared them with those that are known from the known structure?
Yes, I have. Because I'm actually looking at the NAD binding domain, not the protein as a whole. So I compared the NAD binding domain from this GAPDH to other NAD binding domains.
And if I may ask, sorry, Taj, if I may just ask from her quickly, based on what you've confirmed, Kagiso, what because if you want to use because GAPDH is also present in humans, you know that.
Yes.
Yeah, so if you really want to make or identify any drug compound that basically will be used in treating an infection in human, how will that discriminate? Because GAPDH is also in humans, so you don't want to actually inhibit the GAPDH in human, and then because you want to target that of the worm. So what is the what did you get when you did the alignment? What is the difference between the two GAPDH, the one of schistosoma and that from the from human?
Oh, it's actually 18%. That is the difference. But then that's why we also try to look at the antigens on the GAPDH from the schistosoma, because on the paper, there was a paper I read where they found that there's actually antigens that elicit an antibody response that the GAPDH from the human doesn't really, sorry, I'm trying to find the word, where the GAPDH of the GAPDH in the human doesn't really elicit that antibody response. So there's actually immunoglobulins that can differentiate between the schistosoma GAPDH and the human GAPDH.
Okay.
Right, I see that Wolf Dieter, you're back again. I hope so. And we have lost you towards the end of the
Yes, sorry, kind of my Zoom seems to have crashed in the kind of just as I was speaking to Kagiso earlier. But I think we've basically discussed the point. I think it's just a matter of Kagiso really looking at the model of the two of the complex and trying to get some supporting information either from pre-existing complexes or by really looking internally whether that really makes sense. And it might very well be that you've got the right complex. It's kind of just nice to have some supporting information about it.
Yes, thank you.
Right, thanks for those remarks, Wolf Dieter. Okay, we still have plenty of time for questions. If there are any other burning questions, we would be glad to accept them. Okay, I don't see any other hands or any other questions in the chat. But please do take note of the fact that you can get hold of all the speakers on the conference website. If you go to the participant's name on it, you should be able to find the email address. So please continue to engage with each other, even after these sessions and after the conference. And this then concludes the 2 sessions of this morning. Let's give a hand to our 2 speakers of this session who have both done a great job. And we are now going to break for an hour and a half, and I hope to see you all back for the next session this afternoon, where we'll have another 3 talks, also on very interesting topics. So please have a look at the conference program, and I hope to see you all back again. So thank you everyone for your participation.
Thanks, Chad. Thank you very much.
Thank you. Thank you, Tat.
Thanks to everybody.
Thanks, Trevor. We'll talk later.